Hello, everyone. I'd just like to say thank you to Microbes NG, of course, for the opportunity to speak today, give a quick flash talk. So my name's Adam Mulkern. I'm from TwinCore, which is a joint institute between Hanover Medical School and Helmholtz Centre for Infection Research. And the title, the very broad title of my work, is The Impact of Genetic Background on Antimicrobial Resistance Evolution. And I'm currently using E. coli as my model. And my hypothesis is that different bacterial strains from the same species might vary in their propensity to develop antimicrobial resistance and follow different adaptation trajectories within the sequence space. So here's just kind of an overview of that work. So we've got lots of E. coli isolates, hundreds of natural E. coli isolates from very diverse genetic backgrounds. We've got lots of different antibiotics covering all of the major mechanisms of action, so about 12 antibiotics. And we're using automated high-throughput laboratory evolution, so with the Opentrons and PINs and Gilson pipettes, to try and passage as many of those strains as we can with increasing selection pressure to determine their adaptation potentials in the presence of these antibiotics, but also look at their adaptation trajectories within the sequence space. So, after looking in the literature, we decided that possibly the best way to apply this selection pressure is using a drug selection ramp or maybe an evolutionary ramp. And what we do is we work out the initial susceptibility, so the MIC value, and then create this ramp going from 1/8 of that MIC value all the way up to, say, 16 times and even 64 times MIC in some cases, over, say, 8 to 10 days, and increasing, doubling in concentration every single day. And then at the very last time point that we see kind of an acceptable level of growth, we take this strain as the adapted strain. So in this example, in the image here, it's at 4 times MIC. And here's some examples from my work. So we've got OD on the Y, and then the extinction step or the MIC step along the bottom here. And you can see, for example, for this EC958 strain in the red line, in ceftriaxone, it's surviving all the way up to 16 times MIC in comparison to some of the other strains, which are dying at a much earlier point. And then conversely, say in tobramycin, they're having slightly different patterns, say, in their growth, and they're becoming extinct at different points. And this is showing that there are differences in evolvability between the different strains and between the different treatments. And here's just kind of like a more formal example of that. So here's preliminary data from 88 strains, showing not only the differences between the strains within the different treatments, but also differences between the different antibiotic treatments. So we're seeing lots of different variation and some interesting examples, for example, like ceftriaxone. And again, I've just included another example, so we go ahead and sequence. So yeah, what we're noticing is hopefully supporting our hypothesis that genetic backgrounds have an effect. So different strains on the left-hand side are acquiring different mutations, so the genes across the bottom. But actually, a lot of these genes, although they are different mutations, they're actually convergent in function. So when you take a look at BAS, OMPR, OMPC, EMBZ, they're all around this kind of cell wall inhibition, which is perfect as ceftriaxone as a cell wall inhibitor. And just finally, with some of the adapted strains, we do a lot of things like fitness assays, but also like cross-resistance and collateral sensitivity assays, just to see if, say, a strain is adapted and resistant to amikacin in this case. So the turquoise dots are amikacin-evolved isolates. They're, of course, resistant to amikacin, but then also resistant to tobramycin as well. So there's this cross-resistance, which makes sense because they're both aminoglycosides. But then even more interesting, for example, these amikacin-evolved clones on the right-hand side are becoming collaterally sensitive to ceftriaxone. And with that, I guess I come to my conclusion. So yeah, I believe that we are providing plenty of proof that genetic background is having an effect, and that different strains within the same species are varying in their propensity to develop antimicrobial resistance. But yeah, we're hoping to provide a lot more proof and get this paper out at some point. So yeah, I'd just like to thank the group of Professor Marco Galladini, my funding body, Resist, and Microbes NG, and thank you for your time.